Liquid biopsies are a revolutionary class of clinical diagnostic tests which have the potential to provide physicians with critical information that can aid in disease diagnosis, prognosis, or for selecting the best treatment options for their patients. The purpose of this video is to explain why genomic DNA modifications are important and how clinical tests, such as the Ivygene test, detect these DNA modifications within blood samples in order to provide valuable information to an ordering physician. Most cells within the human body contain 6 billion base pairs of genomic DNA, which encode an estimated 20,000 genes. In order to function, each cell must be able to properly regulate the expression of each of these 20,000 genes. This means that a cell must be able to increase or decrease the production of each gene product in response to both the cell's local environment and signals received from other parts of the body. Human cells use a wide array of different mechanisms to properly regulate gene expression. One such mechanism involves the modification of the DNA-based cytosine by the addition of a methyl group. Importantly, cytosine methylation is not a mutation that alters the genetic code. Instead, this type of DNA modification acts as a reversible marker that influences gene expression. Some genes contain special regulatory elements. When these regulatory elements contain a lot of methylated cytosines, then the gene tends to be turned off or silenced. In contrast, when these same regions contain few methylated cytosines, the gene tends to be turned on or expressed. In other words, by identifying which of these regulatory elements are methylated, it is possible to gain insight into which genes within a cell are turned on and which genes are turned off. Now, let's take a closer look at changes that happen in cancer cells. When a normal cell transforms into a cancer cell, there are significant changes in gene expression as the cell begins to overproduce genes associated with cell growth. These changes in gene expression coincide with changes to the methylation pattern of the cancer cell's genomic DNA. Therefore, normal cells and cancer cells can be distinguished by detecting which sites within the genome are methylated. Therefore, by determining methylation patterns of tumor or cancer DNA, it is possible to confirm cancer presence. The genomic DNA of cancer cells can be shed into circulation, often due to cancer cell death. This circulating tumor DNA can be isolated from blood samples, and the DNA methylation pattern can be identified by using advanced sequencing techniques. This pattern can then be compared to a database of known DNA methylation patterns of various cancers to aid in cancer detection. The new class of liquid biopsy tests, such as the Ivygene test, that investigate ctDNA methylation patterns, are improving patient outcomes by providing critical information to physicians to aid in disease diagnosis, prognosis, and possibly the selection of the most beneficial treatment options.